This video is going to model meiosis and crossing over and all the genetic variation that happens in meiosis using PlayDoh. So in class you were asked to make sister chromatids, two pairs of different chromosomes, sister chromatids. So here are mine. I've got this nice big chromosome and I've got this little chromosome. Okay, and these are sister chromatids. They are completely identical. Here's my other pair of sister chromatids, completely identical. Then you were asked to make their homologous pair out of a different color. So here's my homologous pair for the big one, and here's my homologous pair for the little one. Okay. So these are homochromos, homologous chromosomes. They're similar, they're not completely identical. Maybe here on this chromosome, there is a gene that says eye color. Well, over here is that same chromosome that says eye color, but the allele for that chromosome is going to be different or could be different. If we're talking about sisters, that gene for eye color is the exact same gene as over here. But on the homochromos, it's a gene, but it's not the same allele. It's not the same version. All right, so we're ready to start prophase 1 in meiosis. And during prophase 1, homologous chromosomes coil up, like get really, really close. Okay, so not only do my sister chromatids form and get close, but my homochromos get real close. This is called synapsis. Synapsis. All right. When these chromosomes are so close together, they can actually cross over. This can physically cross over. My sisters don't want to stay connected. Okay. They can physically cross over and actually break off and exchange genetic information. Well, now this sister chromatid is not completely identical to this one because it gave some of its DNA over here. So this is actually called, real complicated, y'all, crossing over. And this happens in prophase 1, and it is when the chromatids physically cross over and break off and exchange DNA. Now, the point right here, that specific point where they cross over, is called a chiasmata. Right there. Right at that spot. Chiasmata. Okay, so at the end of prophase 1, this is what I've got. Now, likely crossing over happened on these homologous chromosomes also, but for simplicity's sake, we're not going to model that because it's just too complicated and too time consuming. All right, so we're done with prophase 1. At metaphase 1, metaphase 1 is going to look like this and this. My spindle fibers are going to attach here and here and here and here. Okay. At this point, how they line up also leads to genetic diversity. These chromosomes could have lined up like this, to where the predominantly green ones were on top, the predominantly blue ones are on the bottom. Or they could have lined up like this. Okay. So for simplicity's sake, we're going to have the green one up on top, blue one on the bottom. These pencils and highlighters are representing the spindle fibers. Spindle fibers attached to the centromere. All right, in anaphase 1, these spindle fibers shorten and pull the homologous chromosomes apart. In telophase 1, we end up with two different cells. Here's a cell, here's a cell. Notice we didn't split the sisters. We split the sisters in mitosis. We'll split the sisters here in a minute in meiosis 2, but we're not splitting the sisters. So this is what the cells would look like at the end of telophase 1. Two cells, sister chromatids in each cell. Now, how do we count chromosomes? We count them by the centromere. So here's one centromere, here's two centromeres. So there are two chromosomes in this cell. Here, one chromosome, two chromosomes in this cell. Are they the same? No. This one's real big, this one's little. Totally different chromosomes. Now, ploidy, haploid versus diploid. These cells at this point are considered haploid. Why are they haploid? Because you only have one. You only have the paternal chromosome. You only have the paternal chromosome here. You only have the maternal. You only have one of the two copies. So this is like having the left shoe, and here's the right shoe. Left shoe's over here, right shoe's over here. When we started this process in prophase 1, we had the left and the right. We had the paternal and the maternal. Paternal, maternal. But in this instance, let me get this back too. So this is what telophase 1 would look like. All right, prophase 2. Between telophase 1 and prophase 2 is a step called inter-I-N-T-E-R, kinesis. Okay. Interkinesis. And that is just a simple step, a stage, a resting stage between telophase 1 and prophase 2. During that time, these chromosomes would turn back into chromatin. These in this cell would turn back into chromatin. Okay. But there's no copying. We don't make another copy of the DNA. All right, so now we're ready to do meiosis 2, starting with prophase 2. Prophase 2, my chromosomes coil back up. My sister chromatids are all together. Metaphase 2, these guys would line up at the equator. Anaphase 2, these chromosomes split apart. Oh no, we're breaking up the sisters. You have to divide the centromere. You break up the sisters. And so this is going to form a cell. And this is going to form a cell. So at this point, here's a cell, here's a cell, here's a cell, here's a cell. So recapping this whole thing, at the end of telophase 2, we get 1, 2, 3, 4 cells. These cells have one copy of each chromosome. So they're said to be haploid or 1N. And notice, are any of these cells alike? Are they identical? No, they're very, very different. So we get 4 genetically unique cells. And that, ladies and gentlemen, is meiosis modeled for you using PlayDoh.